Je le fais en anglais, je ne serais pas incapable de le faire en
Au
espagnol.
contraire, c'est très bien en anglais. ¿Listo? Hola a todas y todos, bienvenidos por este tercer día de la conferencia de BioPhysImmuno que hacemos aquí en Institut en virtual total este año, porque es la conferencia 1.5. Y vamos hoy a tener dos charlas. La primera de Paolo Pierobon sobre la mechanics and polarity of B cells y la segunda de Keya Sengupta sobre la física de las membranas. I will switch in English to present and introduce Paolo. Paolo works in Paris. He was previously at the Institut Curie and now established an independent lab in the Institut Cochin in Paris. His work is mainly around B cells and the biophysics, immunobiophysics of B cells, from the single cell scale to the more integrated scales of tissues, such as in the lymph nodes, which is most ongoing work. So Paolo today will talk about the mechanics and polarity of B cells, which is a very important step in their function. So Paolo, you can go. You have roughly an hour, including the questions that will be voiced at the end by Diego, who is just next by. Thank you.
Okay. Thank you, Pierre-Henri. Hola a todos. I will switch to English immediately. I won't massacre your language. So as Pierre-Henri said, I'm working now in the Institut Cochin in a group which is called Dynamics of Cytoskeleton-Dependent Response of Immune Cells. We are interested in any kind of study of the role of mechanics for and cytoskeleton in the function of not only of B cells, but T cells and also macrophages and monocytes. I will focus mainly on what I did in the past, I would say 10 years, which is what happened when the B cells encounter the antigen. So B cells, you know, does it work? Yes, B cell, as you know, is the antigen, the antibody producing cells of our body. And they first, before being able to produce an antibody, they have to encounter the antigen. The antigen is any particle which elicits an immune response. And this occurs in the lymph node where naive B cells encounter the antigen under a soluble form, or more often, depending on the size, on the surface of an antigen presenting cells, which is in general the macrophages. So this starts the formation of what is called an immune synapse. Maybe you had already talked about that. So immune synapse is, as an analogy with a neurological synapse, is a platform for signal exchange between two cells, and where you really have not only the contact of two cells, but you have a passage of signals and passage of materials in concrete molecules going from one side to the other. So the antigen in general is presented from these cells in a native form. It means basically the protein against which is going to be formed the antibody is indeed recognized roughly by this receptor, which is a B cell receptor called BCR, and is internalized. So this is what actually, just a second. This is actually what happened when you have, for example, a bacteria entering, or a pathogen entering the body for the first time, and it occurs in this side of the lymph node. This is what starts the production of an antibody. The transformation that the cells undergo after encountering this the antigen, there are many transformations that the cell undergoes. So it can be the cells differentiate afterwards in a plasma cells, which is able to produce the antibody, and the antibody is precisely coming from the same locus where this B cell receptor comes from. And so the B cell receptor is in essence linked to the membrane, but has the same structure or very similar structure to the antibody that is going to secrete afterwards. So this is this contact is what happened. What led us to study the B cells, B cell behavior. It can be mimicked in many ways. To be studied from the biophysical point of view, people observe, for example, the synapse in vitro in this context. For example, by plating the antigen on a cover slip. And you can do very nice microscopy. Like in this case, it's not from my group; it's from Michael group, and Madison Burger Monroe was for a few months in our lab, and she was doing basically this SIM microscopy, super resolution image of the actin structure at the synapse. And this is in our group where we basically plate the B cells, and we are looking at the polarity protein, which is concentrated toward the center of this synapse. And these take advantage of the fact that you have something which is flat and nicely and relatively rigid, and you can actually rebuild a bilayer, like mimic really the surface, the free surface of a membrane. And so this is on supported lipid bilayer, where you essentially have the formation of this microcluster of BCR. Actually, the antigen is fluorescent, and what you see is actually the clustering of the antigen. And this is something where you don't have basically an antigen recognized by the BCR, and so the cell is essentially not reacting. So what is really interesting and stereotypical is this shape of the synapse, which occurs only in this type of naive cells. For the B cells, but it can be found in T cells. Maybe you have seen other type of synapse with the centrosymmetric patterns. So in this way, we are looking at what happened at the cells only at the contact. We are also interested in other type of observation, and this of course, when you want to look at cross section in a transversal section of the two of the cells, or two cells forming the synapse. So in this case, we can study what happened inside the cells. So we noticed a few years ago, if you present a bead which is coded with the antigen, you notice basically that the cell polarized the centrosome, so the microtubule organizing center, which is basically the place from which all the microtubule starts from, and it regulates the traffic of lysosome in such a way that it concentrates towards the formation, toward the synapse. If you coat the beads with a dye which is sensitive to pH, which is this cipher, at a certain point, this basically turned on, indicating a drop of pH, so a secretion of acidic proteases. And we came up with this model, basically, when you engage the BCR, so you form synapse to essentially polarize the cells in such a way that the traffic of the lysosome is actually concentrated towards the synapse, and there is a release of proteases, and these proteases are indeed chopping the antigen and allowing the internalization of the antigen. So this is I won't talk about the process that follows, but essentially what happens is that this antigen need to be processed, load on the major histocompatibility complex, and further in subsequent days, the cell is basically, or hours rather, the cell is basically able to present this antigen on the to a T cells, and this establishes a dialogue between the two cells, which ultimately tell the cells, decide basically the fate of a B cells in terms of becoming, as I said, a plasma cells or a memory cells, for example, and undergo also transformation of this loci recombination, which leads ultimately to the secretion of antibody, which are of a very high affinity. At the beginning, these antibodies that will be secreted in the first wave, they're not super high affinity, but it's the whole process is what permits the maturation of high affinity antibodies. So these experiments were actually, keep in mind these movies, because these are the few movies that we could get in this paper back in 2011, and what leads basically to develop a system to multiplex and to accelerate the process of acquisition, data acquisition in this context. So roughly at the same time where we show that there is the acquisition of antigen through this enzyme release, another group show that in other situation, when you have a deformable substrate like possibly the membrane of real cells, you can basically have a mechanical acquisition. So you really literally the cells pulls in the BCR with the antigen attached on that. And so this leads us to understanding what happened at the interface, and so to measure ultimately forces at the surface. That's where we establish a traction force microscopy assay. So traction force microscopy is a technique which is used to measure adhesion forces, typically in fibroblasts, by putting the cells, splitting the cells on soft substrates like a polyacrylamide gels, where you have fiducial marker. These are basically fluorescent beads, and you measure basically the displacement of these beads from the caused by the forces exerted by the cells on the substrate. We adapted the system in such a way that we are not only measuring adhesion forces, we are indeed measuring the forces that the cells is producing only when you activate the BCR. So we plate, instead of plating just a simple fibronectin or adhesion molecule here on the surface, we plate an antigen, and the cells are much softer and smaller than fibroblasts, and that's basically where we have to adapt our system to have basically very soft gels of about 0.5 kilopascal. And also we adapted the system in such a way that we are not measuring. Typically what you do is that you measure forces at the beginning and at the end after several hours that the cell has been plated. Rather, you detach the cells so that you can take basically the picture with a 10 minutes difference. So you let the cells adhere, and then you just measure the displacement of the cells after you detach the cells. And you take the difference between the two, and from an algorithm, you can deduce the forces that the cells has applied on the gels. But we did something a bit more sophisticated. We investigated in time what happened. The typical movies that you see is that basically once the cells adhere, so we just deposit the cells on top of that, and we have so our relaxed state when there is no cells, and then we see this contraction in time. And we can also use myosin 2 GFP cells. So these are primary, by the way, murine cells. We can track the signal of myosin 2. And myosin 2 is the model which is responsible for cell contraction. So what you can see is basically that you have some collective contractions which correlate very well with the arrival of myosin 2 GFP. So what is interesting is that some of these beads are indeed not moving collectively as you would expect from a typical elastic medium, but they are basically jiggling around without being too correlated with the rest of the beads. And so we split the components of the displacement in coordinated and non-coordinated one. We show that non-coordinated indeed are more present in the center of the synapse and correlates with the position of actin patches that we showed indeed that there were actin protrusion. At the same time, myosin, you would expect that to pull on the antigen, that myosin is necessary because it's something that creates the contractions, and so you would have expected to have the position where the antigen is internalized correlating with the myosin dot. But the myosin is more, if you average on time, you will see that it's basically more concentrated at the periphery of the synapse, and this correlates pretty much with the pattern of collective forces, indicating that there is this force patterning that is correlated with the two different actors of the cytoskeleton. And myosin 2 is probably not necessarily related to the site of antigen acquisition. The site of antigen acquisition correlates more with these patches of actin. But if you switch off or you inhibit the myosin contraction using a drug, using blebbistatin, you lose these antigen, these active patches. You lose also the contractility. and you lose the antigen extraction. I'm cutting the long story short because we basically set up an assay to measure the antigen extraction by having essentially a fluoro which is quenched close to the gel and dequenched when it's internalized. But you can see basically that if you use blebistatin, you're essentially impeding antigen extraction and impeding contraction, but impeding also the presence of these actin patches. On the other side, if you activate myosin contraction using a drug which causes a release of calcium or lysosomal calcium at once, essentially you reach the same level of antigen extraction, but in half of the time. And indeed, the energy, the contractile energy that you can measure are actually double. So the model that we came up with is essentially explained that we require myosin 2 for collective contractions and for the formation of actin patches. So if you remove myosin 2, you lose essentially the actin patches. The actin patches is where the antigen is internalized, but the myosin 2 is acting probably from the periphery, pulling or not. Our argument is that the myosin 2 concentrates all the machinery necessary to the internalization of the antigen in the center of the synapse, where the real internalization occurs. And the force we measure at the periphery are indeed not sufficient to justify an extraction of the antigen, because we are way below a few piconewton per receptor. Someone else measured the single molecule level an extraction force of something on the range of 20 piconewton more than what we estimate simply by dividing the stress by the number of receptors. So it's possibly that there are all the myosin implicated or all the mechanism for the precise localized antigen extraction. So as you could see, there are many ways to present the antigen. So we have essentially systems which are relatively fluid. Well, we started with the polystyrene beads, so the system is not fluid and is not deformable. I show you some investigation with the belayers. This allows to have a mobile, to have an antigen which is actually mobile, but the system is not deformable. And indeed, in all these cases, cells polarize and exert. They polarize towards the antigen, and they don't internalize the antigen in a mechanical way. We are measuring forces using deformable gels, which are not really mobile, but at least they are deformable. We can measure forces, and we ask ourselves if we could combine mobility and deformability in a single substrate, and that's where we could start collaborating with Giacomo Taccioli, chemists at the École Normale Supérieure, and we had the common share students setting up a system of droplets. So lipid droplets are indeed fluid, so they allow the synapse formation very similarly to the way belayers would do. These are lipid droplets which are coated, which can be coated with phospholipids. In general, they're biotin-native phospholipids, and to which you can basically link via streptavidin a biotin-related antigen or an antibody eliciting the response of the cells. And so this, the advantage compared to beads is that they are deformable, and so basically by tuning the surface tension, essentially we are tuning the rigidity of these objects, and the chemistry and the fluidity are to some extent well controlled, or anyway better controlled compared to a polystyrene bead, in the sense that the droplets, because they are lipids, are essentially extremely can be functionalized in a very specific manner. And the fact they are deformable indicates that we actually could use them as a force sensor. So that's is more a proof of principle that we could see only quite rarely. And so basically we have no statistics, but I can show this movie at least where we see some deformation. So the B cell is arriving here, and this droplet is actually deformed. It's also deformed in another piece because there is a system holding the droplets here. And the stress you can get is basically obtained simply by measuring the curvature difference from a sphere. a measurement in surface tension, which is a measurement you can take in bulk. And this is simply Laplace law is what regulates the stress in an inflated balloon. We also set up a new system to measure polarization, because we were, as I said, the movies, the polarization movies using the beads were quite rare and very difficult to acquire, because we have to wait for the instance where the cells encounter the beads. So you can do it with very low magnification. It doesn't, it's not impossible, but then you miss a lot of subcellular details. So we have these microfluidic chip that allows at the encounter to multiplex the encounter of the observation of two spherical objects, which could be two cells, or like in this case, droplets and the B cells. And you can, of course, flash drugs. You can control the genetics of the cells outside and just use the cells you want inside. We adapted both for primary and cell lines. I'm going to show you from now on mainly cell lines. Typically, what you do when the cells and the droplets encounter, so here there is these microfluidic traps, is that the cells basically polarize. So these cells have huge nuclei, and you see the centrosome, so the microtubule organizing center, moving towards these droplets. And you can also see in these droplets the accumulation of fluorescent antigen at the synapse. And the students actually characterize very well how many molecules are indeed necessary. And we estimate about 75 antigen molecules per square micron. The saturating basically concentration in the droplet, surface of the droplets. And so Lea, the PhD students, work in modifying many properties of these droplets, changing the oil, changing the surface tension, changing the density of the antigen, and to some extent we can also change, by changing the way we functionalize the droplets, the fluidity of the system. So we can investigate. We could observe, for example, that if you can drag the antigen under the synapse, you can basically be activated antigen if you have a fluid droplets. So I'm not going to touch this part, which is a little bit technical. You can read if you want the details. You can ask about the protocol in this paper. And what we did also with this kind of droplets was actually to study the polarity of B cells by looking at several compartments, and that's basically the aim of another PhD that we supervise, who has as aim to establish a sequence of events that was leading to polarity. And that was Judith Pinot. So she took many movies in 3D in time of a sequence of polarization using, for example, fluorescence actin, and establishing when the actin was arriving, for example, at the synapse compared to a non-activating surface, and establishing essentially the maximum of the time of the recruitment time to characterize the sequence of events. So at the beginning, you have a formation of a synapse through actin. Roughly at the same time, you have the signaling signal peak. The DAG is the acylglycerol, which is basically a fluorescent reporter for signaling, for one of the early signaling occurring at the synapse, which is slightly after the formation of the actin. The centrosome is probably the most robust indicator of polarization. You see, it's amazing how little noise you have in this polarization dynamics, and this was done using either fluorescent centrosome or using sear tubulin, which is stained for microtubules, which is drug actually is Taxol, but at a very low concentration, you can actually mark mainly the centrosomes where the microtubules are very concentrated. So that's why you see some signals of the microtubules. This occurs in about 4.5 minutes. Golgi apparatus essentially follow the centrosome dynamics. The lysosome are a bit particular because they actually polarize, but they're also secreted. So at a certain point, the polarity is they are recruited, but actually they go away pretty early within the first 10 to 15 minutes. And the nucleus is quite particular because the nucleus seems to indeed move around and then or reorient and then move back. For this, we have to do another kind of analysis. We basically segment in 3D and we track this indentation. So in such a way that we could establish an axis of polarity intrinsic to the nucleus. So this indentation is tracked as a minimal local curvature of the nucleus, and as you can see, we can basically decompose a rotation movement and a translational movements. And in a way, we can see that at the beginning, basically, the nucleus is the center of the nucleus is staying where it starts, so basically about in the center of the cells. And after 10 minutes, essentially when it has rotated, so this is the angle of rotation, which is stable at around 30 degrees within 10 minutes. After that, the nucleus is basically moving back. And so we could observe that, and we could link this to a depolarization of a detachment, actually, of the microtubule organizing center from the nucleus. So the mTOQ is basically driven towards the synapse, and the nucleus is actually pushed back. And we are talking about a few less than microns scales. It's really very small movements, but at the scale of the cell is actually important. And something which we haven't understood yet is this flattening out of this indentation. So it seems that the distance between the center and the place of the indentation is systematically increased with the activation of the cells via BCR engagement. So this is still a question that we have. And so it could indicate that the nuclear shape is actually linked to the genetic program that is actuated in the cells once you activate, because the cells afterwards mature and becomes a cell which is expressing then presenting the antigen and deciding whether to become plasma cell or memory cells. So it seems that the B cell polarity is basically forms in two phases: an early and local phases, where essentially everything plays a role, stays at the synapse, and the later global cells, where basically the microtubules play a role. So we play with these with two drugs that are perturbing the actin. So latroncolin A is depolymerizing the actin. What we observe is that the DAG is basically reduced and the antigen recruitment is reduced. So this is the control that I showed you before, and this is the latroncolin. As you can see, but if we depolymerize the actin, essentially the microtubule organizing center is already detached from the nucleus and is very prone to polarize. So it's actually polarized quickly. The polymerization is indeed maintained. So the process is actually faster. And the nucleus shows no rearward motion because it's already on the back, essentially. It's on the other opposite, because there's no link between the microtubules and the nucleus. So it's not really particularly interesting. What we found more interesting is actually when you perturb the microtubules. That's the second drug we use, nocodazole. So in a control phase, you have in control experiments, you have essentially polymerization of the polarization, sorry, of the nucleus. The nucleus seems to be kind of keeping this bean shape. But when you have the nocodazole, the nucleus looks like a massage, and the cells is like moving overall in direction in space. So we have several geometric indicators for that. A variance of the aspect ratio, the solidity, which is essentially the indicator of how the cell, how rough is the contour of the cells. And you can see that in the nucleus you have this increasing in the change, and this change in the aspect ratio when you depolymerize the microtubule. So this frequent change in cell shape and in nuclear shape prevents indeed a correct synapse formation, in particular because the actin now is not anymore concentrated to at the synapse, but is indeed but shows patches everywhere. And if you look at the antigen recruitment, this is relatively fine, and the signal is also is actually more sustained. But if you measure the actin maxima, you find many more patches of actin around the cells, and which are at the distance, which are actually far away from the synapse. So they seem completely decorrelated, as they were forming many synapses. So we dig into the literature, we find out that when you depolymerize the microtubules, indeed you are releasing a GAF. So a GAF is a molecule that activate small Rho GTPases, which are proteins that control the polymerization, sorry, of actin or actomyosin contraction. In the particular case, it has been shown that the depolymerization of microtubules can lead to release of GFH1 and the activation of Rho-A. Rho-A on one side lead to formin-mediated actin polymerization, and on the other, ROC-mediated myosin 2 activation. So we start playing with the GFH1. We noticed already that if you form a normal synapse, you can have a concentration of GFH1 at the synapse, an enrichment of GFH1 at the synapse, and if you disrupt with no codon, you get basically GFH1 concentrated in other places, and you lose this enrichment basically at the synapse. So indeed you free somehow GFH1 by disrupting microtubules in these cells as well. So if you silence GFH1, we have what we expected. Essentially, the geometry of the synapse is kind of restored, and also the geometry of the cells. At least some of the parameters we could look at, we used to characterize the geometry of the cells, like the solidity aspect ratio, are indeed recovery, indicating that if you switch off this release of GAF, we have somehow a good synapse, and we have and we do not perturb basically the shape of the actin as much as we do when we depolymerize the microtubules. So we play also with the Rho-A. So by using a dominant negative Rho-A, we can see that we essentially recover the same parameter as if you haven't used basically the no depolymerization drugs. And on the other side, if you use the constitutively active Rho-A, we get basically something which is very similar to what we had if we depolymerize the microtubules. And we also play with ROC by showing that essentially most of the actin patches are mainly due to forming, while the contraction are mainly due to myosin 2 contractility, as we expect. So what does it mean? What is the biological implication of that? Because in theory, we don't have normally the microtubule depolymerization. But what we find out is that the BCR is indeed has been shown to acetylate the microtubules, and the acetylated microtubules is indeed leading to a GFH1 release. So indeed, by depolymerizing microtubules, we are essentially exaggerating this action everywhere, by the way. So we are basically localizing the GFH1 everywhere. So we interpreted our data in the light of what is called the LEGI, so which is local excitation global inhibition model, which is the simplest model for which you can explain phenomenologically polarization, the formation of polarization axis is used in chemotaxis, is used in cell motility, in morphogenesis. And we use it in our system. So we looked for local excitation and global inhibition. So the LEGI model states very simply that if you have something which is locally, if you have a polarity Q which is locally excited, which means whatever polarity, because it means it could be myosin 2 or the concentration of a molecules or a certain chemical reaction. In our case, it could be essentially the acetylation of the microtubules that leads to the GFH1 release. And if this process is inhibited everywhere in your cell, in your bacteria, these two conditions are enough to maintain a stable polarity axis. So why do you need the deacetylation? The global inhibition? Well, we need global inhibition because otherwise the local excitation would spread very quickly, and this axis wouldn't be an axis anymore. So there are small short differential equation showing basically that you need these two conditions, otherwise the system wouldn't be stable. So we look for our global inhibition, and we thought there might be some deacetylases that are actually acting on the microtubules everywhere in the cells. And these deacetylase can be actually inhibited by using drugs, and we show that you can basically reduce to some extent the GFH1 enrichment at the synapse by inhibiting indeed this deacetylase. So by switching off the global inhibition, we are indeed losing some properties of this polarity axis, and also we are basically destroying this asymmetry between acting in the front and in the back. It's not extremely clear, but we have other arguments. We have other data that shows the action of this deacetylase. So in particular, we show that what could happen is that the system is indeed try to maintain a single polarity axis. You can actually form two polarity axes and two synapses, which looks roughly the same. And these occur in particular in no-codazole, so in these depolymerized tubules cells. Why? Well, if you look at cells forming multiple synapses, within 10 to 5 to 10 minutes, the cells tend to bring all the droplets in the same places, and actually to form a single polarity axis where all the antigen is indeed accumulated on one side. If you depolymerize the microtubules, the fact that you have acting everywhere shows that you can form synapses, but these synapses are completely separate. So we counted the number of synapses that you can form. For this, we cannot use our microfluidic chips, so these are just bulk experiments, so we don't have the dynamics, but we just counted the percentage of cells forming one or two synapses, and we see this percentage is indeed increased. The one forming two synapses is indeed increased when you depolymerize the microtubules. And the percentage is recovered if you depolymerize microtubules when you silence the GFH1. And if you disrupt the deacetylase, so the global inhibition pattern, you increase the frequency of, you favor multiple synapses, because you are indeed favoring the presence of multiple polarity axes, and you're breaking the Legge model basically. So this is also true when you have when you measure motility. These are short experiments that we didn't characterize very well, but we could show that basically the straightness of the cell motility in vitro. So these are cells that are I forgot to put the movie. It's really nice. You have cells that basically are forming the synapse with the droplets, and then they are dragging the droplets around, and also the mean directional changes. So essentially, the straightness of your cells are both impaired by this. If you depolymerize the microtubules, basically the cells doesn't have an axis anymore, and it doesn't know how to move. Okay, so in conclusion, we have these two classes of events: the actin base and the microtubules base. And the microtubule destruction amplify a process which is occurred in general through BCR stimulation, which is basically the release of GFH1. And the destruction impacted the formation of a single axis, while in a normal situation, the local acetylation of the microtubules favored the GFH1 release and the establishment of a single polarity axis, thanks to the control of the local actin polymerization. So in a nutshell, the actin and microtubules are basically cooperating to stabilize a single polarity axis for these cells. And this, we think, is actually a mechanism which is much more general than only the B cell synapse, basically. Okay, so every time I explained this, at least in the past, the question was, is this a real thing? So, or just a fantasy? The idea was that indeed, our cells polarizing, our cell forming this kind of synapse, our cell exerting forces, and why are doing that in vivo. So in vivo, the situation is much more complicated. The immune cells are actually moving, the surrounding cells are moving, so they are actually bumped. They bumped in the neighboring cells, and they are in a very fluctuating environment. And the stroma network is also moving and perturbing the motion of these cells. So this is a very old movies by Frohnangen labs, where you see B cells in red capturing the antigen from macrophages. It's a capsular cynosome, and you see that these cells are actually moving around and bumping on each other. And all the black that you don't see is mainly stroma and possibly other cells. So the situation is much more complicated than in vitro. So this pushes us to go to a more realistic systems. Also motivated by the fact that people will start looking at the geometry of the lymph node and realize, for example, that during inflammation, we know since probably 5,000 years that when we have an inflammation, our lymph nodes can eventually swollen. And this swelling is actually leads to an increase of tenfold the volume of this organ in the space of few days. So there's a question how you accommodate the stress. There are measurements that have been shown that show that the stress is indeed released thanks to a relaxation of the actomyosin network at the single fibroblast level, and how all this is regulated and how all this impact or not the encounter of the antigen with the by B cells or the synapse by T and B cells. This is really what actually drives our research now. So the project we propose, and it has been fun and still ongoing, and is basically the center of our interest now, is to characterize the tissue rheology and to perturb this tissue mechanically or chemically, and to link this to something which you can control in vitro in the system that we have, namely the traction force microscopy, where you can actually measure forces while you try to disrupt by shear flow a synapse, or the microfluidic traps, where you can actually favor the synapse with a compressive flow, favor also a synapse which is normally shouldn't occur with something which is non-specific or low affinity anyway. And the idea is basically to play with this with the different with what we observe in vivo and what we observe in vitro. In vivo, we are not really doing as anybody would as an immunologist, a proper immunologist would do intravitreal microscopy, because you cannot perturb, and it's very difficult to measure tissue rheology. So we are taking advantage of a system which has been developed by a previous researcher. The researcher was previously working in Institut Cochin, and namely the lymph node slices. So we are doing basically we are cutting in slices this lymph node, and we keep them alive for a few hours. So they are 300 micrometer thick slice, where you can do this kind of imaging. And the nice thing is that we don't need any genetically engineered mice, so with anything fluorescent, because we can stain the cells we want with the color we want. And so if you're interested in the CD3 compartment, CD4, CD8, or subcapsular sinus macrophages like we do here, and B cells, we can basically do it in situ. And keeping this slice alive in a biosystem that I will show in a minute. So this work, this is the job, basically the work of Luisa Burons, who actually third-year PhD students in my group that set up all this work, and basically did the measurements that I'm going to show briefly. I'm trying to keep I try to just to give an overview, also because we don't have a very deep data on a deep understanding of the data, but we are setting up, and we basically, I'm going to show you basically the protocol a little bit. So Emanuele Donadio started this study for mainly immunological reason. So you basically harvest the lymph node from mice and you embed it in agarose. You cut them, you slice them in 300 micrometer small slices. So the lymph node, just to give you an idea, is about, imagine a grain of rice of about 1 millimeter times 2 millimeters, a small ellipsoid, basically 1 or 2 millimeters size. And so the slice can be immunostated like we did here with the antibodies for the T cells and B cells, or in the subclassical macrophages, just to know what I showed you before. And the imaging is done at 37 degrees with a perfusion of oxygen. And we are now working on a microfluidic system, which should in principle increase this observation time. So what we can observe now is the motility of the cell, but it's very difficult to track these cells. What we wanted to do is actually to characterize the activity of this tissue by means of an external tracer. So that's where we use our lipid droplets. This time they are lipidol droplets, which means it's heavier than water. So we can basically just place them on top of our slice and we get them inside. So what you typically see is this motion of these droplets, which is induced by the motility, the local motility of the cells. And some of the droplets, that was our initial aim, is that are actually fusing and forming a very large droplets where you can basically see some deformation. We haven't characterized yet all this. Probably the real perspective is actually to micro-inject very big droplets, but we probably have to use another oil with a lower, with higher, surface tension, because the one we use basically just squeezed through all the cells, basically. So what we can do now is basically tracking these beads. We guarantee that they are indeed inside, and we can basically track them using TrackMate algorithm. And what we did so far is indeed characterizing their motion by observing the mean square displacement. So the mean square displacement is essentially how far the particle went within the given time, and is calculated on each trajectories. And typically, when we're talking about the diffusive motion, this exponent is indeed 1. So there's a linear relationship. Mean square displacement grow linearly with t, and we can characterize the diffusion coefficients. If it's a thermal diffusive motion, this diffusion coefficient is related to the viscosity of your system and to the essentially to the viscous coefficient of the droplets. But here we're not in a thermal environment, so these droplets that you see are actually moving differently in a different zone, and they're actually moving because they are pushed around by the different cells. So we are nevertheless using a diffusion coefficient as a proxy to understand that. So the typically look very linear at the beginning, and at a certain point they start moving out from these regimes. But here the problem is that we don't have enough data to be sure, and so we will perform longer time experiments. We compare two different situations. We compare a situation where we could stain the T cells using a CD3 antibody, which is known, which is basically dimeric. And we know that when it's dimeric, it's indeed activated T cells. When you activate the T cells becomes less mobile, so they actually stop, and you reduce the global activity of the tissue. So we used these two systems to show that basically, we use the antigen, which is actually dimeric, and we also transform it by an enzymatic process with using peptin. We crop this antigen having monovalent Fab fragment, and this is supposed not to activate the T cells, and T cells are actually in this case more mobile. And indeed, you can see this difference in the diffusion coefficients. This is just the distribution. We hope we could see it in the MSD, but as I told you, we are not very. This noise here is indeed very big, and we cannot really observe for a long time because the track are indeed quite short. So we started doing some experiments. Is only n equal 1 for longer time. If you observe the slides for longer time, short time you recover with the same diffusion coefficient, but at a certain point you start seeing after a few minutes the droplets, the MSD flatten out. This would indicate that droplets are indeed kind of caged, trapped between different cells, but after a certain time, which is about 10 to 15 minutes, maybe 20 minutes, which is a bit longer than what we could observe in the previous movie. We see the cells, basically, the droplets taking up again a kind of diffusely or super diffusely, we haven't been able to characterize yet, movements, which is not the case indeed when you have the activated antigen. So this would indicate that the droplets, if you wait long enough, are indeed able to, they're pushed out basically of this cage provided by the cells. So this could be an indicator for the activity, although you already see some indication of the activities of the cells at very short time, because the diffusion coefficient is roughly an order of magnitude higher. And we can, as you might have noticed, that the zone in the lymph node are not the same, not behaving the same way. The B cell zone is much more packed, and they don't have, they have a stroma network is actually different. So we segment them based basically on the staining of the cells. And what you could see is that the B cell zone have a diffusion coefficients, the droplets in the B cell zone have a diffusion coefficient which is much smaller compared to the T zone, about more than an order of magnitude. And now we can basically apply the same principle to different lymph nodes. So what we start doing is basically a model for cellular inflammation provided by a collaborator, a team of collaborator, which is a mouse model for rheumatoid arthritis that show is a knockout for a kinase. It's called 6-3. And we are interested because we can observe a lymphadenopathy, which means essentially that the lymph nodes are super huge. And actually, the shape is also different. They form secondary follicles. The geometry is completely different, and we are still characterizing that. But what is interesting is that essentially the diffusion coefficient is extremely smaller, and also the behavior of the MSD is extremely different. You see in the knockout, essentially, the cells, the droplets are barely moving, and then they get trapped essentially in the first 2-3 minutes, while after being trapped in the wild type mice, basically they are kind of free after several like 10 to 15 minutes. If you look at the number here, we haven't understood why, but they are essentially all 10 times higher, which is probably due by the fact that we are becoming better and better in using this system. The system is extremely, I'm not trying to oversell it, is extremely variable. So you're working with live tissue, so it's they are very delicate, and there is a big difference between the movies that you can take essentially right after you dissect the mice and the movie you're taking 5 hours later, for example. So this is something we are still in the process of characterizing. And as a perspective, we have probes to look live at the collagen. And so the correlation between the motion of the cells and the deformation of the collagen is something that is actually very interesting, and that we can hope to link to the not only to the motility of the cells, but to the activity, intrinsic activity of the tissue, to measure in particular in situation where you have an inflamed lymph node, so similar to what we have in the knockout that I showed you before. So in conclusion, we have a system that is actually a proxy for cell activity, and what is interesting and what it provides this system of slices that is something that you cannot do in multiphoton microscopy, namely an open system that you can actually control. So you can put any kind of color, any kind of drugs on top of that, and also you can perturb by compressing it. So we start doing that. We're still like try to find out the optimized way not to kill the tissue after 10 minutes. But indeed, it is possible to perturb it a little bit, to compress it, to observe what it's doing. Just simply, even the genetic is already interesting if you do bulk transcriptomics, for example. And there are a number of things that we have to test, like a microinjection to be larger droplets, which is really challenging, and perturbing the environment with the real immunizations. What I show you is a sterile immunization. It means there's no pathogen. The inflammation, sorry, sterile inflammation. So the system is inflamed without the presence of a real pathogen. What we want to do is actually put in something that activates the cells because is pathogen is generating immunogenic, is generating immune response. And so the idea would be to immunize mice basically and observe what happens at different time point to the mechanics of this tissue. And I'm just advertising the PhD of a student that just got recruited. She's working on these measurements in tumor draining lymph nodes and measuring the difference between an inflamed lymph node versus a metastatic one. And so this is basically what the direction we are moving right now. And with this, I would like to thank all the people that collaborate on these projects, from past collaboration, present collaboration, and people of my group in particular. Anita and Judith and Leah, they were the 3 PhD students that were very important for the beginning of the for the slide I showed at the beginning, and Luisa Bruno, this is finishing her PhD, that did all the setup and the measurements in the lymph node. And I would like to thank you for your attention. Ready to take question. Thank you very much.
Thank you so much, Paolo. I first have to say that really impressive work. The technical depth of your work is truly, truly impressive. And not just me saying, one of the comments is, thank you very much for the excellent conference. Measuring these forces at the nanoscale is an enormous challenge. You have shown us some brilliant techniques. So
I'm very happy. Thank you.
And of course, I have to say that we definitely need to meet and collaborate, because we have some of these interrogations, and we have some systems in microfluidics. And of course, we would love to put in place some of the techniques you're showing.
Definitely. So I let Pierre-Henri do all the job so far, but I'm happy. By the way, I don't know if I put my email address, but I think Pierre-Henri can provide it.
Then so you have to visit us in Mexico, and then next year.
Okay, I will come. But if students want to contact me, or a researcher interested to contact me also for postdocs and so on, I think it's possible to set up collaboration in the near future.
Thank you. And then we will take some minutes of our next speaker to do some questions. We have one
sorry, Kaya.
No. What are the main limitations of the techniques, such as the ones you use for capturing the full real-time dynamics of the forces patterns in a synapse, and where should these technologies be headed?
So I would say that the real limitation is that we don't know what these forces are for. So we are measuring forces through, we're measuring forces applied to a substrate via the BCR. So in general, if you ask, for example, people doing torsion force microscopy for the fibroblast, they are able to do that. In general, they are able to trace them to focal adhesion. And so they can actually analyze the structure of focal adhesion and what happened there. Here, the system is much smaller. The cells are much smaller, and so the control of the forces is not really super clear. We don't know if what we see is the forces that are transmitted because the cell is pulling on the BCR, or because the cell is, for example, rigidifying simply and changing the structure inside, and we see basically just an effect of that. There are other techniques, but they are not really combined with these techniques. For example, to observe the pulling of the BCR at a single molecule level. So I think one perspective would be actually to combine the two. So to combine, for example, a force sensor at the molecular scale, for example, with DNA gauge tension. So they're basically DNA that are programmed to break at a certain force, or to open at a hairpin that are supposed to open at a certain force. This combined to larger scale measurements like the one of the attraction force is probably something which is interesting. There are works done mainly on T-cell synapse where you have a more controlled 3D measurements by super resolution of this kind of forces. So these are work by Marco Fritsche, for example, in Oxford, where they can basically increase the resolution that we have. Still not going to the, I mean, it's difficult to say if it's really due to the pulling on the receptor, or because we are pulling, because it's difficult to say if the cell is pulling on the receptor, or we are observing a deformation of the cell through the pulling of the receptor. So this, I think, is the main limitation. Okay. And Pierre maybe can complete, because he worked also a lot on the
in his seminary.
In fact, just to say that this is the coupling between the two traction force microscopy methods is the center of the topic we put for the European network. So that's something we are going to push, because indeed there's many applications where you may need the molecular to cellular transition levels. That's the only way to observe the coupling.
Thanks, both.
Technically, it's not so easy.
Observing, but the structure that we see, the actin structure that we see, we try to characterize using vinculin, paxillin, a bunch of molecules which are in general related to podosomes structure, something like that. The only analogy that we found is indeed what is called invadosome-like protrusion, which was proposed for T cells precisely in the process of diapedesis. So the structure we see in the 3D protrusion is indeed related to, I mean, the structure that might use the same components, but this is not absolutely done for diapedesis. The cell is encountering. I show you the picture in the movies in vivo, where the cell is basically scanning, patrolling the tissue, and at a certain point encountering the antigen on the surface of the macrophages, and basically getting the antigen from there. And then the time it spends, so that depends pretty much on the quantity of antigen and on the affinity. But this is not diapedesis. But there are some components which are actually used.
Okay, we have two questions. And then one is, given how inflammation alters lymph node, mechanical and stromal tension, do you think this mechanical perturbation could destabilize the single polarity axis in B cells and promote multiple synapses, similarly to what you observe with microtubule acetylation or excessive GAF-H1?
FH1, yeah. So we hope so. That's why we proposed the project, but we haven't. I can't answer yet. But we hope that basically by inducing a perturbation, we can actually recapitulate some of the process that occurs during inflammation, and so we can basically control establish what is mechanically driven and what is completely cytokine basically determined.
Okay. Then we have your use of droplets as passive mechanical reporter is fascinating. I'm agree with that.
Thank you.
Do you think this approach could be extended to map local anisotropies or gradients or stiffness within the lymph node, essentially turning droplets like microbiology probes? Or?
Yeah, that was originally the idea. It turned out that it's super difficult to microinject a lymph node and a lymph node slice. Because essentially the lymph node, what people have been doing, for example, J Campus and in Dresden now, and other people. and the droplets inside, or you use the polyacrylamide droplets as a force sensor. What OJ was doing is actually micro-injecting zebrafish, which is more difficult, but still the zebrafish is transparent. The lymph nodes are not transparent, so we don't know what you're doing. And when you approach the micropipette, basically you break it half of the time. You don't know where you're injected, basically. And when you inject, what we find out is that the oil the surface tension is so low that the oil basically kind of penetrate everywhere and squeeze everywhere, and there's no droplets anymore. So that's a problem. So we need to change we change oil, and possibly change also the approach on the way we micro-inject it. So far we are going from the top, and probably we need something which is different. So we are working with the platform of in vitro fertilization, for mice in vitro fertilization in our institute, because they're actually the most expert of this in this process. But that was the original idea, to have an internal sensor. You also need to produce a larger droplet. These droplets are the scale of the cells. What we need to do something like that is on the scale of 5 or maybe more cells. So something which is 50 micron or 70 microns large. So it's a little bit different.
So, well, thank you so much
thank you.
for having you here. And of course, the audience is about 20 people. And I think it's a really good audience, because we are in the Faculty of Science and we have different interests. But thank you so much.
Okay,
and
thank
then
you.
We'll pass to Kayan.
Thank you, it was a pleasure. Bye.
Will present. Bye.
Thank
Thanks.
you, Paolo. Bye. We keep in touch. So just for let you know, if you want to look for the email of Paolo, you should look at his name and Institut Cochin, Cochin in Paris. So our next speaker is Kaya Sengupta. So Kaya is a physicist, and she's now leading the physics of living system section of the nano and micro technologies microsystem and microtechnologies in SINAM in Marseille. She did a lot of work on lipids at the very beginning of her career, including substrates with supported bilayers, but also giant vesicles work. And all of this, in fact, is very linked to what we are interested in, because the membrane is the first mediator of cell contact to the exterior, and is one of the first hub for the signal gathering. So Kaya proposed to have today a small talk-like lecture about the physics of this membrane, that is very often underlooked, because we always think in terms of more integrated systems. But the membrane is really the key place where everything starts when two cells are contacting. So Kaya, if you are ready, then the floor is yours. Thank you.
Okay, thank you, Pierre-Henri, for this nice introduction. I assume that you can hear me. All right. So the talk was not supposed to be, you said, a short introduction. Yeah, I mean, for an introduction, it's short, but I normally prepared for about an hour. You can stop me when you like if I'm talking too much. So I'm going to talk to you about membrane physics, biophysics. And I usually like to give this lecture with lots of questions, but unfortunately, I won't be able to hear your answers, but I hope that you would answer nevertheless. Okay. Do you see the slide changing?
Yeah, okay, thank you.
Okay, great. So the plan is the following. I'm going to talk about membrane and life, then membrane components and architecture, phase transition and criticality, diffusion, transport, etc., elasticity, curvature, etc., and adhesion. I must say that this is not a lecture where, in the sense that I really teach you about membranes. What I'm going to do is to tell you what are the important things about membrane to learn, and then you can teach yourself. Okay. I'm not going to talk much about transmembrane transport, and I'm absolutely not going to talk about electrophysiology. Membrane and life. So who knows why I have this picture here in a slide about membrane and life? I assume somebody put up their hand and you can answer to yourself, but to answer that question, let's think about what is life. What are the minimum ingredients of life? So, as you must have, learned in, I don't know, middle school, living things, they eat, that is to say, they consume energy, they grow, and they die. That's not enough, right? Because crystals also grow, and you can say if they redissolve, they're dying, right? So we say, okay, living things have to reproduce. That is to say, create duplicates. But imagine you have a crystal, you break it into small pieces. Each piece can act as a seed for a new crystal to grow. Well, is the crystal now living? Of course not. So there is one crucial element missing in the story, which they don't necessarily tell you about in middle school, but I guess you know by now. This is evolution. So evolution is the change in heritable characteristics, right? And of course, you all know that our evolution is, like we are able to replicate ourselves because of these replication molecules, and evolution happens because of mutations. But if you think about it now, evolution needs compartmentalization. Why? Because imagine that in the primordial soup, when life began, let's say we had some RNA which are coding for some protein, which gives you some advantage. If this RNA lives in solution and shares this advantage with all other RNAs, then there will be no evolution because there will be no selection. So for selection to happen, for evolution to happen, there must be compartmentalization. So the thing is that people who work on origin of life, they more or less agree about when it happened, so 4 billion years ago. But there's still no consensus about exactly where it happened. So more and more people believe that life originated in deep sea vents, hydrothermal vents like this. That's why I put the picture. And there is I don't know how many astrobiologists or early life biophysicists actually agree with this picture, but there is an idea that not only did the chemical ingredients, so the first replicating molecule, the first metabolic molecules, not only did they develop in here in the hydrothermal vents, but actually the first cells, these were pores in the hydrothermal vent, inorganic pores, and they showed that in fact you can have proton gradients and therefore create energy if you have, holes in rocks like this. Okay, you don't have to believe that life began inside as pores in rocks. But what you can, of course, understand that in order for life to leave this and populate the earth, you needed compartmentalization. You needed compartmentalization in order to make cells, in order to make evolution. And so one imagines that, these are our ancestral protocells, which are leaving this hydrothermal vent wrapped in a membrane, right? So what are these membranes which wrap all cells today, which form the wall of all cells, and they must have formed the wall of the protocells as well? Now let's take a step back and ask ourselves, what is the ideal envelope for protocells? It's an engineering problem, right? Give yourself 2 seconds to think, what are the attributes which are needed? You need an exchange with the outside, so it must, which is controlled. So this must be semi-permeable, right? Fully permeable doesn't make sense because you need some selection. You need surface transport. You need it to be soft and flexible so that you can change shape. You need control interaction with the environment. It would be nice, if one could pinch it off in order to divide into two, etc., etc. And what is the best material to do that? It turns out that the best material to do that is exactly a lipid bilayer. Okay, so which is basically the cell membrane, which is a two-dimensional, highly heterogeneous liquid in three-dimensional space, right? Okay, so let's look at this membrane in detail. What are the membrane components and what is the membrane architecture? So, when I knew I had to give this talk, I asked an AI bot to make a picture of membrane for me. And it correctly made this picture where you already see that there are lipids, there are cholesterols, there are proteins, some weird ones, but there are proteins, some of which are transmembrane. And there is the glycocalyc sugar, which you see here. So everyone knows this, even an AI. Let's go deeper. What are lipids? So we'll take all of this one by one and go through them. So what are lipids? Lipids are essentially molecules which look like this. They all have a head, which is the green and the red part. The head is connected to chains, which are the pink part. And this is connected by this yellow part. Very interestingly, the yellow part can be either an ester bond or an ether bond. And all across animal and plant kingdom, mushrooms, most bacteria, what have you, we all have ester bonds. But there are some very weird protea which have ether bonds, which make people think that, cellation happened 2 times, that 2 times life left these hydrothermal vents wrapped in 2 different kinds of lipids. Anyways, so these are lipids. They self-assemble to form a bilayer, and we are going to talk about this later, self-assembly. The second, so lipids, they look like this, they are named like this, DMPC, DPPC, etc. And what does that mean? So this PC basically is referring to the head group here, which you saw in red and green here. So PC means this head group. You can have a different head group, then it would be called, let's say, PE or PG. This part of the lipids name, this refers to the chain. So 14-0 just means that there are 2 chains with 14 carbons and there are 0 double bonds. And then 18-1 means 18 carbons with 2 double bonds on 1 on each chain, etc., etc. And these guys make up 50% of the lipids on our cell membrane. So here is the zoo of other lipids. You have different head groups, PSP, I talked about PC, PI. The PIs, there are very, very few of them in few copies in each cell, but they still have enormous impact on metabolism. Okay? Then there are these phengomalins, which are weird guys on the glycolipids, which have long sugars on their head. Okay, let's talk about cholesterol. So cholesterol looks like this. A lot of people call cholesterol a lipid, but it's not a lipid because a lipid has a head group and a chain. Cholesterols, when you bring them to a cell membrane or membrane of whatever kind of lipid membrane, they would actually go and sit right here, mostly with the chain and a little bit at the interface. And what they do is to disrupt the structure of this lipid membrane and do very strange things. They increase diffusion, so they increase the fluidity of the membrane. lipids can go faster, but they also increase the stiffness, so the membrane is less bendable. And they also increase the stability of the membrane, making the membrane less fragile. Proteins. So there are a huge number of proteins associated with the cell membrane. They can be of different kinds. They can either just be, lie close to the cell membrane and interact with it through charges, or they may have bits which are inserted in the bilayer. They may be attached to a lipid. And here you see a whole zoo of possibilities of transmembrane proteins. The last component is sugars. So, which are the sugars of the glycocalyx? And you see some cartoons here and the electron microscopy image here. We know very little about, compared to proteins, we know much less about the sugars, but because they're very difficult to, image. There are many techniques to tag proteins, but not that many to tag sugars. And yeah, I have written here inner versus outer, because what you need to appreciate is that because of presence of the sugar, the inner and outer leaflet is quite different, but also because the lipid composition of the inner and outer leaflet are quite different. There are a lot of proteins which you saw, which should be facing either the outside or some which should be facing inside, etc., for things to work correctly. Okay, so all these are important things to keep in mind. Now, the lipids. It turns out that the shape of a lipid or a molecule similar, which is also an amphiphilic molecule with hydrophilic head and hydrophobic chain, depending on their shape, they make different structures in water. So if I have a lipid which has a roughly cylindrical shape, it will make a bilayer. But if I had, something like this with a profile which is conic, it makes a spherical object which is called micelle. So a spherical micelle. With a different shape, you have all these other different possibilities. So this I put here from a general perspective of soft matter, that lipids needed to be this shape in order to form bilayers. And this is just an idea of if we had different shapes, so surfactants are capable of forming all these different kinds of assemblies. Today, it's important to know about these assemblies because some of these even weird assemblies like this one, are used for delivering of drugs, delivering of mRNA, etc. Okay, so what I told you so far is that membrane chemistry, so the shape of the molecule, etc., it couples to its physics, meaning the way it self-assembles. Now let's spend a little bit of time talking about phase transitions. So I'm sure all of you know what phase transition is, right? The transition from liquid to gas, liquid to solid, etc. These are phase transitions. In membranes, you also have phases. There are phase transitions in membrane which does not disrupt the bilayer structure, but it changes the bilayer properties. So the cell membrane, our cell membrane, is usually in this phase, where the chains are molten. What does molten actually mean? It just means that the carbon-carbon single bonds, they are free to rotate. So since each carbon-carbon bond is rotating, essentially the chain can change conformation. As a consequence, the lipids themselves are able to diffuse. And if you look at the head groups, just the head groups, you will see that they are completely random. This is what you see here. each one, like each of these V's represent one chain. Now, if we go down in temperature, then the chains, they freeze. And they freeze in what looks like a crystalline structure. This is very ordered. And this is called the gel phase or L-beta phase. However, this is not a crystal. If you look at the head groups, they are still random. How is this possible? This is possible because there is only one head group per two chains. So the chains are in the lattice, okay, you see that? But which chains are actually connected by a head group, this is still random. And then there are some weird phases, such as what is called the ripple phase, where the bilayer becomes corrugated. And of course, if you go very far down in temperature, then you get a true crystalline phase. Okay, so we have been talking about, so these phases have been seen in what I call membrane stacks. So they are recapitulated in a single membrane, but a lot of this was learned from stacks of such bilayers. Now, you can appreciate immediately that this is unrealistic, the membrane that I show you. Why? Because you see a lot of yellow. The yellow are the chains. The chains are hydrophobic. And so hydrophobic means water hating. And if you put such a membrane in water, it's not going to like it. Its energy is going to be too high. So the membrane has to do, the molecules of the system has to do something to hide the yellow from the water. What can it do? What it can do is to close up and form something like a balloon. So this is what a vesicle is. It's a membrane which is closed in itself with water inside and water outside. And these I will come back to these vesicle systems later and to show you what we can learn from these simple cell-like systems. So this is like a cell, except that inside you have nothing, outside you have nothing. You just have a synthetic membrane. Okay, the phase transitions I talked about, this happens in pure lipid systems. If you have mixed systems, then you can have some more interesting things. For example, if you mix sphingomyelin with DOPC, which you saw before, and cholesterol, which you also saw before, then you can have two fluid phases, one which is called liquid ordered and the other which is called liquid disordered, and you can imagine why from the name, right? This is more ordered and this one is more disordered. But these are both liquid and different from the gel phase that I talked about before. So solid ordered phase, meaning this is really gel phase or completely like crystalline. Liquid ordered phase, where these are moving, and liquid disordered phase, where the chains are also slightly disordered. And of course, these are both liquids. Now, there is a very interesting idea, which is not new, but maybe something like 20 years ago, people started realizing that the cell membrane is, in fact, at a critical point. So if those who have done basic physics courses, you will recognize that this is the water phase diagram, where you have solid, you have liquid phase, you have vapor phase, and then you can go to the liquid phase from vapor phase either by passing a phase transition line, or you can have supercooled vapor and then go bypass that. And this point is called the critical point, the star, right? People realize that if one extends this analogy to membranes, then people realize that the membrane is at the critical point. And it's at a point where it could either phase separate or not phase separate. Okay, so this is the experiment. You have different kinds of lipids which are perfectly mixed. And as you slowly increase the temperature, you get a phase separation. So by changing the temperature, you see the numbers 24.7, 24.3, 4, 2, and up, you have a phase separation. Now, the point, why is this interesting? This is interesting because for years, people believed that membrane has what are called rafts, which is like phase-separated domains. But it turns out today that membrane does not have rafts like people imagined before, large domains, but they have very small domains, which are always dynamically forming and deforming. And there's a very interesting experiment where this was Sarah Veach. She separated the membrane of a cell from the cell itself. So it made the cell bleb, take out the bleb, and then looked at at what happens to the lipids. And very quickly, they phase separate. So the cell is doing something to keep the lipids all mixed up all the time. Okay. So the membrane has very dynamic nanoclusters. How exactly these nanoclusters are formed and what exactly is their role, this is still a topic of research. Now, for these clusters to form, one mechanism could be what is called hydrophobic mismatch. What does it mean? It means that if I have a protein with a transmembrane domain, which is slightly larger than the lipid bilayer thickness, then the bilayer will be forced to deform. Similarly, if it's slightly smaller, it will still go in the bilayer, but the bilayer will be forced to deform. The chains will deform. And because of this, proteins of similar size would like to stay together so as not to create too many of these deformations. So this is called, this thing is called hydrophobic mismatch, and it may be a way for protein clusters to form in a very generic manner without having specialized protein-protein interactions. Okay. Now, this is important also for formation of, insertion of transmembrane proteins into the bilayer. So here is an unfolded protein. If it can fold like this in a thick bilayer, which is, a thicker bilayer, which is matching its final size, then this is happy. But otherwise, there is a problem. Okay, so the sizes have to match correctly. Okay, so that's all I'm going to talk about for this scale. So I'm not going to talk about membrane simulations and such like. And in case you're wondering what the martini bottles are doing here, this is because martini is a very famous way to simulate things like what I showed you just now, insertion of proteins. And very briefly, what it realized is that so the problem is this: if you want to simulate a bilayer and you want to do atomistic calculation, you can imagine that there are all these atoms on the membrane. On each lipid, there are lots of atoms, but there's also the water. So that's a lot. So what people realized is that we could perhaps replace the details, the atomic details, by simply spheres. Okay, so this is called coarse graining. And thanks to coarse graining, one can now simulate huge membranes like this. Okay, let's go now to diffusion. What is diffusion? Diffusion is basically the movement of a particle buried within a system, and this particle moves simply due to thermal fluctuations, due to thermal energy. So if I think of diffusion of, let's say, an ion of sodium, so a sodium ion from salt that you dissolve in water, then this moves around because the water molecules are fluctuating and kicking this sodium atom around. All right. The same thing happens in the membrane. Each lipid is kicked by its neighbors. Each protein is kicked by all the lipids, right? So that's why diffusion happens. And it's so, okay, so the diffusion is usually quantified in terms of the diffusion constant, which is given by this equation. So what is this equation telling me? The equation is telling me that, okay, I start from a position which I call x0, and then I look at the position xt as time progresses, so t is time. I calculate this difference. So basically, this is the distance, the vectorial distance. I square it and I average it. So mean squared displacement is proportional to time, and this proportionality constant is 2 times diffusion constant. All right. So this mean squared displacement, so this quantity, okay, it's proportional to time only when the diffusion is normal, which is only because of thermal fluctuations and in absence of too many barriers. If you put too many microscopic barriers on the way, then you get what is called subdiffusion. The mean square displacement changes like this. And if you don't, and if you are like kicking actively, for example, if in my water I had bacteria which were like kicking my particle, you can have super diffusion. Now, the diffusion of anything, but also proteins, right, is given by what is called the Langevin equation, which is nothing but balance of force. So you have the inertia here, mass into acceleration. Then you have friction. So the friction coefficient times velocity. Then you have an elastic force, which is proportional to your displacement. And this is the random force, which is there because of thermal fluctuation, the thermal random force, okay? So you equate all this. This goes away when we are talking about small things like proteins. And this is all that we are left with. And this friction term is related to the diffusion constant by the famous Einstein-Smolchowski relation. Okay, so this Einstein relation is nothing but how diffusion is related to transport. Okay. So what can you do with this? What you can do is infer activity. What do I mean? I mean, to know whether something is, let's say you're given a glass of cytoplasm and you want to know whether the cytoplasm is still active. All you have to do is to follow the motion of, let's say, whatever organelle you can look at. And if its motion is, if its mean square displacement is linear with time, it means that there is no activity. So people were doing this with bacterial bath. And so this is quite interesting to look at active matter. Okay, we'll come back to this later. Okay, so diffusion on a membrane is, this D is given by what is called the Sackmann-Delbrück model. You may come across this, and the main thing to remember from this is that the diffusion constant goes as the logarithm of 1 over the size of the particle. And this is basically telling you that you can have small lipids or you can have large proteins. The diffusion is not that different. It goes as the log, okay? And then what you need to remember is that if you are close to a wall, then things might be different. So here I just plot, just to show you how slow log of 1 by x is. So 1 log of 1 by x compared to x, this is quite flat compared to this. Right? Okay. So the implication is, so what Safman-Reinbook model is telling you is, number one, that the head group size really does not matter. All the dissipation is coming from the transmembrane part. You can put as much huge cargo as you like on the lipid. Diffusion will not be very much affected. The other thing that I wanted to tell you is that double bonds that we saw earlier, these disrupt the chain order and they increase diffusion. At a given temperature, you better have more double bonds in order to have more diffusion. Cholesterol increases diffusion because, like I told you, it disrupts the order. Proteins move slower than lipids because their transmembrane part are larger than lipids, but not by that much. So size of transmembrane domain is important. Then structural complexity is important. So there are length scales, like, small things move faster, but then there's also activity. You can, if you put active forces, you can of course make them go faster. So free diffusion versus active transport. Close to a wall, there may be stronger coupling than log. And then you can also have diffusion not in the plane of the membrane, but across the membrane. Okay? But I'm not going to talk about that. That's transport across membrane. So let's think for a few minutes about numbers. How fast is lipid diffusion? So if the diffusion constant is, this is around that for lipids, typically 1 micrometer squared per second. And then you can calculate that a lipid will go over the entire surface of a bacterium in 10 seconds. And in lymphocytes, this is 5 minutes. So that's quite fast. How fast can a protein be dragged with the 1 piconewton force? Again, you go back to the Einstein equation that I showed you. The diffusion constant for proteins is roughly 0.1 micron square per second. If you put that in, you will see that a single Marsin motor exerting 1 piconewton force, it can pull a protein with the huge velocity, millimeter per second. That's enormous. Snails go at 3 millimeter per second. So, this is macroscopically large. That's mind-boggling. And if you think of, typical DNA origamis like 100 nanometer by 100 nanometer, and you assume that all of these, like all the lipids which are under this anchored DNA origami on a surface, they are pulled with the origami, you will find that the origami is still diffusing quite fast, 0.3 micrometer square per second. So large things can still diffuse. Okay, so since this is a lecture, this is homework. Please verify the numbers, and Pierre-Marie will check your homework tomorrow. If you scan this, then you can get the questions. Okay, that was a joke. All right, now to elasticity. How should we think of deformation of membranes? So if you think of cell deformation, all the forces and stuff that Paolo was talking about, you really need to, if you really want to understand that at a deeper level, you need to worry about how the membrane is deforming when the cells exert those forces, they change shapes, etc. So how can we deform a membrane? Here are all the ways in which you can deform a membrane, and we will go through it one by one to see what is important. Let's start by stretching. So here what you see, these little circles, these are the lipid head groups. I'm looking from top of the membrane, from the above or below, whatever. So stretching, isotropic stretching, it just means that you increase the space between lipid molecules. You try to pull the head groups apart. You can do it. It's quite stiff, but most importantly, it will just break. This will make the membrane extremely unstable, because when you increase this distance, then water is going to enter. And of course, when water enters into the hydrophobic region, this is catastrophic. This is problematic. In that way, lipid membranes are sort of like paper. If you take a piece of paper, you stretch, it's quite difficult to stretch because it's not very stretchable, but it's simply fragile. It's going to break. Okay, let's look at the next one. The next one is shear. What does that mean? It means that in the plane of the membrane, you apply a torque. Okay. Now the question is, can the membrane sustain this shear? The answer, of course, is no. Why not? Because the membrane is a liquid in the plane of the membrane. It's a 2D liquid. And a basic definition of liquid, if you ask like a child, what is the child going to answer? What's a liquid? A liquid is something that flows. And what does that mean? It means that if you try to shear a liquid, it does not sustain the shear, it just flows. You cannot shear a liquid, it will just flow. A solid, on the other hand, you try to shear it, it will resist with the elastic force. Liquid will resist, but with the viscosity, not with the elasticity. So no shear. Now let's think of compression, not the opposite of this stretching, but isotropic compression also coming from the sides. This, of course, is possible. Like, this will give you the bulk modulus of lipids, but it's not very interesting for us because, first of all, it's very high. Secondly, one never has this kind of sideways, and like isotropic compression is very rare. It's not relevant for cells. Okay, now let's try to shear the other way, not in the plane of the lipid, but between the two lipid layers. Then what is going to happen is that the lipid is going to flow again. That's also not very interesting. What is interesting is when you try to bend it. So this bending is very important in lipids. This gives rise to an elastic constant, which is called the bending elastic constant. So when you bend a lipid, it gives rise to the bending energy or curvature energy, which is given by, so the curvature energy per unit area is written like this. Okay, it's inversely proportional to the radius. So basically, it's proportional to the radius of proportional to the curvature, which is 1 over the radius, and this is the proportionality constant. Okay, you don't normally have to worry about this term unless you're making holes. So let's say we are not making holes in the membrane. Let's stick to this elastic energy. Those of you who are following still and not sleeping, you will immediately see that the consequence of this kind of function is that you see there is a R square sitting here. So that means that whatever the size of a spherical vesicle, the cost of making it is always the same. Because this is R square here and it's per unit area. So when you integrate over the area, the R falls out. So the elasticity is proportional to the mean curvature, and this bending constant, bending elastic constant, the bending modulus, this is of the order of kT, 10 to 100 kT. kT is Boltzmann constant and temperature, which is basically the thermal energy. This is the energy that you can pull out of the environment just because of thermal fluctuation. okay? So thermal fluctuations become very easy because the membrane is so soft, okay? This is the point. And then you can have proteins which are curved, which can curve the membrane. If you have polymers which are accumulating, this can also curve the membrane. And these are just the principal curvatures, like the curvature for different shapes. Okay, a few force and energy numbers. Bending modulus, we already said. So a KT is about 4 piconewton nanometer. Then the force to pull out one lipid is about 50 to 100 piconewton. And then if you convert this to work done, what is the work needed to pull out the lipid? This is about 50 kT. So that's not enormous. Huh? The force needed to, so just compare this to force needed to break a hydrogen-hydrogen bond. This is 4 piconewton. But the carbon-carbon bond is 1,600 piconewtons. So that's enormous. And this is what I mean is a hydrogen bond, not the bond between hydrogen molecule, atoms in a molecule. The force to pull a lipid nanotube, on the other hand, this is quite low, 10 to 40 piconewton. Okay. Okay. I have about 15 minutes. Okay, good. So now if I want to write down the free energy of the membrane, because, you know, the physicist among you, you remember that in order to find the equilibrium state of a system, you have to minimize the free energy. So to do that, you need to know what the free energy is. So the membrane free energy is a sum of the bending term, which we just talked about here, okay, this bending energy, plus a tension term, I will tell you what this is in a moment, plus a potential term, which is basically telling you what is the interaction between this membrane and any other surface which is close by. Now, what is this tension term? The tension term is basically related to the tension of the membrane. Very intuitively, you know that tension is basically related to stretching. When I pull the membrane, I'm introducing some tension. Now, with membranes, you can, similarly to polymers, there are two tensions. One is really what we intuitively think of tension as tension, is that you take a paper, you try to stretch it, and you tense it. This is the, or you take a spring, you stretch it, you actually deform it, and that's a tension. But in membrane, since it's fluctuating, there's a lot of hidden area here. So when you try to pull the membrane, you will just straighten out these creases, okay? So that's the entropic part of the tension, and then there's the stretching part of the tension. Now, if you do all that, you will write down an equation similar to this. I will not, you know, from far, it's difficult to explain all this, so I will not talk about this anymore, but just tell you that in cells, the actin elastic energy actually dominates because the actin is much more stiff than the membrane, and the tension is a consequence of actin plus membrane composite. However, if you are looking at phenomena such as endocytosis, exocytosis, blebbing, etc., where you are very, where the actin, F-actin, like filamentous actin, is not a major player, then you really need to worry about these things. Okay, in the last few minutes, very quickly about membrane adhesion, which is my research topic also. So how do cells adhere? They adhere through their membrane, right? And in order to adhere, I don't need to tell this audience that they have these molecules, like these proteins, these ligands and receptors on their membranes, which form chemical bonds, right? What is interesting about biochemical bonds is that their lifetime depends on the force that is applied on them. So in a chemical reaction, you have reactant A reacting with reactant B and making a product. And there is a rate of forward reaction, there is a rate of backward reaction. And if you just, once the product is formed, if you just wait, at some point or the other, it's again going to dissociate. The time for which this stays is called the lifetime. And this lifetime depends on force. So basically, if you pull on this, if you pull extremely strong, it's going to break immediately. But if you pull softly, then it's not going to break immediately, but its probability of breaking will go up. Now, cell adhesion, like membrane adhesion, is complicated, not just because the cell is complicated, but because the chemistry is complicated in such a geometry. Because this is confined geometry, each binding event is not anymore independent. So there's a violation of law of mass action that you learned in chemistry. The forward binding rates are modified. But then, in a membrane, the membrane wants to fluctuate. It's pulling on these bonds. So the backward reaction rate, unbinding rates, are also modified. Now, the earliest ideas were like really long time ago. This is before integrins were discovered. People were thinking that cells adhere through electrostatic or van der Waals forces. And in fact, this happens to be true. When people did measurements, they of course saw that, there are these forces, but in fact, in cells, these forces are hidden by the glycocalyx, and the adhesion takes place through really proteins. So these are some of the earliest experiments whereby looking at the shape of the membrane. So this is a vesicle, which I told you about before, which is like a cell, but with only water inside and water outside. And by looking at the shape change as this vesicle is adhering, you can calculate actually the energy of interaction. Now, that is one way to measure adhesion. One could do better. One could measure adhesion locally by looking at membrane fluctuation. Okay, so how shall we look at membrane fluctuation? The membrane is tiny, and these height differences are also tiny. So those of who were there for Gaurav's talk yesterday, you will recognize these images. So basically, when you need to measure very small distances, very small separations, those of you who did optics, that one should be doing interference, which was, discovered many, many years ago. And there was Adam Curtis, who first used it for cells, but cells are very complicated optically. This is difficult to do. We have done it now, but what we started with was applying the interference to simple objects like colloidal beads or vesicles. So this is what membrane looks like in this interference technique, which is called RICN. So what you see in black and non-fluctuating, these are adhered zones, okay? So basically, this is the experiment. This is the fluctuating membrane. I come with the microscope from below, and I'm watching this membrane. And by looking at the level of gray, I'm able to make what I call fluctuation maps. So this is a height map, and this is a fluctuation map. So this is a vesicle, which is, so let's look at this one. The system is, integrin on the surface, RGD on the membrane, and then they form bonds, which may form domains which are dense, like you see here, 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 and the height. Or it may be sparse, which you see, for example, here in RICM, they don't show up as dark, but these are domains where fluctuations are calmed. Okay, so we think that these are dense, sparse domains. And then, then one can have also very small domains which are not visible in RICM, but which show up in fluctuation maps. If you want to know how to do RSCM properly, you can look at this old paper where we have really explained very nicely all the nitty-gritty of how to do RSCM experiments and how to prepare these fluctuation maps, how to prepare these height maps. This is the height map you see in nanometers, so you're really one can measure with really nanometric precision. Okay, so let's go back to membrane adhesion. I have the elastic term, the tension term. So this is the membrane free energy I told you before. Now that we have specific adhesion, we need to add the bond energy, and then we need to add the entropy of the reactants and products. When you have strong and abundant binders, then the equilibrium is determined by balance of adhesion and elastic energy. But fun begins when you have weak and sparse binders. Then the balance is determined by enthalpy and entropy. And then the dynamics becomes a reaction diffusion problem. Okay, so what is the consequence? The consequence is basically that you get domains simply because of this membrane physics. You don't need any specific lateral interaction. So what is happening here is that if I have the membrane coming down and forming one single bond, there is a lot of force on this. This breaks immediately. But if one happens to form a patch of bonds quickly, then this is stabilized. So we actually did those experiments, and we saw that according to how far the two interfaces are and how fast, how much is the fluctuation, one can have these different types of domains. Either you have, one single one which grows, or you can have multiple ones, depending on the balance between how fast you break the bonds and how fast you form them. Okay, so I did a pseudo simulation to show you this. And basically, if you now have bonds which there are lots of them together, then they are not going to break. Okay, so there is a nucleation which depends on concentration, and this depends on diffusion. So this is a slow nucleation, and it's hindered by the gap, so the distance between the membrane, but it's helped by the fluctuation. And this is followed by a reaction-dominated fast growth, which is helped by the fact that the membrane is already bound, so the next bond can form very easily here. Okay? And now the question we are asking nowadays is, so all this was with thermal fluctuations. This is vesicle, nothing is alive. And we are now asking, what happens with active fluctuations? Do they modulate adhesion? The answer, of course, is yes. You heard this talk from Gaurav yesterday. He used many of these ideas, but in his case, there was the glycocalyx, which was preventing the adhesion. And yeah, but the ideas are really the same. I have another project where what we are doing is to use vesicles still, but fill them with active matter. Okay, so while searching for this talk, I learned that active matter is a video game. That's not what I'm talking about. What I'm talking about is something like cell extract. We do it from Xenopus egg, and then the cell extract is active. We encapsulate it within the vesicle, and which hopefully will give rise to active fluctuation of the membranes, and then we will see what that does to adhesion. So this is a very new project, and hopefully next year I have something to tell you about. Okay, so I'm on the last slide. I want to conclude by saying that, membranes are not easy. Even from a purely soft matter point of view, You have polymers here, you have colloids, you have amphiphiles, and membranes are like really in the middle where they have properties of all these different kinds of soft matter. And I want to say that equilibrium physics is ubiquitous, it's everywhere. Biology has to live with it, or it has to fight it actively, okay? Or use it. So in biological systems, if you are doing physics, then you need to think about this. And last quiz, you can scan these two. Who are these people and what do they have to do with membrane? I would highly encourage you to do this because these are really, really cool stories, especially this lady. Also this gentleman, he was an American politician, but he also was a scientist. Okay, so I would like to end by thanking all, all my collaborators. I have this T-cell work, which I didn't talk about at all today. I quickly told you about my membrane work. And my main theoretical collaborator, Anna Smith, with who I did the work that I presented to you. And I finish with a quote from Primo Levi and pictures that I liked last year when I was visiting Mexico. Thank you.
Thank you, Keya, for this beautiful lecture. I think that we all need to understand what happens in the cells and, of course, in the membranes. And fortunately, we will have it in YouTube to review each time we need it. And I do have some questions already in YouTube for you.
Okay.
So why does the diffusion equation only consider protein size and not some other attributes as the shape or...
Okay, so what is important is the friction ultimately. What is the frictional coupling of the membrane with the object which is diffusing? So if the protein has a very weird shape, what would basically happen is that it will drag some lipids with it, and its effective size, if its effective footprint, will change. And then again, we are back to talking about size. That's the reason we talk about size and not about shape, because at the end, it's the same.
Thank you so much. And another question is, hi, Keya. So, which model allows us to describe the diffusion of a molecule through a porous medium? For example, a membrane.
Right. So diffusion through pore is much more complicated, and I purposely did not talk about it. So I'm not going to answer this question. I'm going to encourage you to look it up, because at the end, it's of course, the same Einstein equation it's fundamental. This one. So a variation of this will describe all diffusion, but it depends on how you adopt it.
Thanks. And then thank you. Well, another comment is thank you for sharing this masterful debrief with us. And I have other questions. In many cellular contexts, the membrane is locally crowded or strongly coupled to the cortical actin mesh. In your view, what is the most limiting assumption of the classical Saffman-Delbruck model, and which modern extension best capture protein diffusion in this non-equilibrium?
That's not an easy question, because there's a lot of controversy in the field about that, and this is, active ongoing research. So even how exactly is this coupled? I have a picture of that. Where is it? Okay, I lost it now. So people, there is the story of the raft, right? There is the story of the actin. Yeah, I wanted to show this. So there is the view that there are cortical actin mesh which coral the membrane. Here, this picture from this paper, what they're saying is that there are tiny pieces of actin which form aster and which actively push some membrane proteins together to form nanoclusters. Okay, so so the question was which model one should consider. I think it really depends on your system. You need to find out.
Okay, and then you mentioned phase transition and near critical behavior in membranes. Do you think cells actively tune their lipid composition to stay close to a critical point? And if so, what experimental evidence you find most compelling?
So cell membrane has to stay fluid for sure, right? So let me talk about that first. So the composition is actively changed in order for the cell membrane to remain fluid. So if you take cells from the leg of a reindeer, these animals which live in the Arctic, so depending on whether this is close to their hooves, which are, sometimes buried in snow, or further up, closer to their knee, the amount of double bonded lipids actually changes. So one has more lipids which will resist freezing in cold further down. This is the same with lipids in fish compared to cold sea fish and tropical fish. So cells do actively control the composition of lipids of the composition of their membrane. Now, what do they do to stay close to critical point or not? This is again a research question, because even this idea of criticality is not something that everyone accepts, and there is still work going on that. And indeed, one of the ideas behind trying to do this encapsulate active matter in a vesicle is to see what it will do to the phase transitions.
Okay, thanks. And then the last one, nanodomains of GPI, anchored proteins, seems to rely on active cytoskeleton process for their lifetime and special organization. How do you reconcile the classical equilibrium description of membranes with these active energy-consuming rearranges?
No, I think there is no reconciliation here, right? Because this is not in an active system. We don't expect it to be in equilibrium. Like I said, equilibrium is important because it's there. As soon as you take out the activity, the system is going to go to its equilibrium state. So it's going to phase separate, and people have seen that. As soon as you take out the cell membrane from the cell, there is phase separation of proteins, there is phase separation of lipids. So, so the point is that the activity is doing something to avoid that, most probably. That's my view, but this is something that we are trying to prove.
So, thank you so much, Kaya. Hope to have you here next year.
I hope so too, and thank you very much. It was very nice to see you, Tatiana. And so all these questions, I'm very happy that I got so many very interesting questions. Thank you.
So for the people connect, thank you so much. We have almost 20 people connected. Wow. And then see you tomorrow at the same time for the last two talks at 10 o'clock. And thank you for being here and do all these interesting questions.
Thank you, and see you tomorrow. Bye.